Welcome to a video presentation of the final project titled Genetic Algorithms for Advancing the Form and Control of Multipurpose Robotic Hands. The code structure looks like this. We have a genome, a phenome, and then a specimen class, a simulation, and an evaluation, and some other helper functions. So we can start by running the tests. We have a test for each component. So while the tests are running, we can open the genome class. There are two genome encodings for fingers and brain. It's just a random matrix of the rows for number of fingers and a column for number of phalanges. And the brain is also a matrix as explained in the report. So the test is run. We had 18 tests in total for the whole components, and every test passes. Start by running test specimen. This specimen would be just the specimen created by the random genome encoding. There was no evolution and no evaluation run. So we can see here a random specimen trying to grab the objects but not succeeding. Let's see another random specimen. You can see some finger movements, but there is absolutely no performance. Okay, with this, starting from this random specimens, 4 iterations were run for 50 generations of 100 specimen, population of 100, and there were results. So let's see the results of the first iteration. For now, we see the results with the training objects. So here it is, picking up the cube, dropping it in the target location, the sphere, and the cylinder. There's one specimen of the first iteration, the fit specimen. Here's another fit specimen performing the function, but as you can notice, some of the fingers are not moving, like they're not performing any function. They're moving in the other direction while the others are actually picking up the object. But still, the specimen was successful in picking up the objects. This one was not successful with the cylinder. There is another specimen. These are all for the first iteration with the training objects. So if we see the same specimens that we just saw for the test objects, which are a bottle and a cup, we can see the first fit specimen successfully moving both the bottle and the cup. There is we can notice a little difference in the finger like trajectory structure, but here they were not successful. For example, this one was not successful in transferring the test objects. This one neither. While this one could transfer the bottle and the cup. So this was for the first iteration. So let's see the third iteration, specimen from the third iteration for the training objects. Here it is, picking up the specimen. This specimen has considerably more fingers and phalanges. And let's see with the test objects. It's the same specimen, picking up the bottle. Finger trajectories, movement trajectories change a little depending on the object. Here the simulation is slow because the cup has more vertices. The 3 object. Okay. So, and now the fittest of all the iterations is the iterations, the specimen in iteration 1. Here, after a lot of runs, there are more fit specimen with interesting finger trajectory movement. So let's see for the training objects. Here we can see the fingers grabbing the objects and more tightly than the previous specimen. And if we see the same specimen for the test objects, we can see some interesting behavior. So let's see the bottle. There is some structure, but when it grabs the cup, it imitates the human finger, like passing the fingers through the cup opening. We see that again here. So these are the results for the project. Thank you for watching.